T helper cell, Wikipedia audio. The T helper cells are a type of T cell that play an important role in the immune system, particularly in the adaptive immune system. They help the activity of other immune cells by releasing T cell cytokines. These cells help suppress or regulate immune responses. They are essential in B cell antibody class switching, in the activation and growth of cytotoxic T cells, and in maximizing bactericidal activity of phagocytes such as macrophages. Mature Th cells express the surface protein CD4 and are referred to as CD4 plus T cells. Such CD4 plus T cells are generally treated as having a predefined role as helper T cells within the immune system. For example, when an antigen-presenting cell expresses an antigen on MHC class II, a CD4 plus cell will aid those cells through a combination of cell-to-cell interactions and CD40L and through cytokines. CD154, also called CD40 ligand or CD40L, is a cell surface protein that mediates T cell helper function in a contact-dependent process and is a member of the TNF superfamily of molecules. It binds to CD40 on antigen-presenting cells, which leads to many effects depending on the target cell type. CD154 acts as a costimulatory molecule and is particularly important on a subset of T cells called T follicular helper cells. On TFH cells, CD154 promotes B cell maturation and function by engaging CD40 on the B cell surface and therefore facilitating cell-cell communication. A defect in this gene results in an inability to undergo immunoglobulin class switching and is associated with hyper-IgM syndrome. Absence of CD154 also stops the formation of germinal centers and therefore prohibiting antibody affinity maturation, an important process in the adaptive immune system. Activation of naive helper T cells. The importance of helper T cells can be seen from HIV, a virus that primarily infects CD4 plus T cells. In the advanced stages of HIV infection, loss of functional CD4 plus T cells leads to the symptomatic stage of infection known as the acquired immunodeficiency syndrome. When the HIV virus is detected early in blood or other bodily fluids, continuous therapy can delay the time at which this fall happens. Therapy can also better manage the course of AIDS if and when it occurs. There are other rare disorders such as lymphocytopenia which result in the absence or dysfunction of CD4 plus T cells. These disorders produce similar symptoms, many of which are fatal. Following T cell development in the thymus, these cells recent thymic emigrants egress from the thymus and home to secondary lymphoid organs. Maturation of root in slow results in the generation of mature naive T cells, but naive T cells now lack or lowered the expression of the root-related surface markers, such as CD31, PTK7, complement receptor 1 and 2 and the production of interleukin 8. Like all T cells, they express the T cell receptor, CD3 complex. The T cell receptor consists of both constant and variable regions. The variable region determines what antigen the T cell can respond to. CD4 plus T cells have TCRs with an affinity for class II MHC, and CD4 is involved in determining MHC affinity during maturation in the thymus. Class II MHC proteins are generally only found on the surface of specialized antigen-presenting cells. Specialized antigen-presenting cells are primarily dendritic cells, macrophages, and B cells, although dendritic cells are the only cell group that expresses MHC class II constitutively. Some APCs also bind native antigens to their surface, such as follicular dendritic cells, but unprocessed antigens do not interact with T cells and are not involved in their activation. The antigens that bind to MHC proteins are always short peptides, 8 to 10 amino acids long for MHC class I, and up to 25 or so for MHC class II. Type 1 hypersensitivity includes common immune disorders such American Samoa asthma, allergic rhinitis, eczema, urticaria, and anaphylaxis. These reactions all involve IgE antibodies, which require a Th2 response during helper T cell development. Preventive treatments, such as corticosteroids and montelukast, focus on suppressing mast cells or other allergic cells. T cells do not play a primary role during the actual inflammatory response. It's important to note that the numeral allocation of hypersensitivity types does not correlate to the response in the Th model. Type 2 and Type 3 hypersensitivity both involve complications from autoimmune or low-affinity antibodies. In both of these reactions, T cells may play an accomplice role in generating these auto-specific antibodies, although some of these reactions under Type 2 hypersensitivity would be considered normal in a healthy immune system. The understanding of the role of helper T cells in these responses is limited, but it is generally thought that Th2 cytokines would promote such disorders. For example, studies have suggested that lupus and other autoimmune diseases of similar nature can be linked to the production of Th2 cytokines. Type 4 hypersensitivity, also known as delayed type hypersensitivity, are caused via the overstimulation of immune cells, commonly lymphocytes and macrophages, resulting in chronic inflammation and cytokine release. Antibodies do not play a direct role in this allergy type. T cells play an important role in this hypersensitivity, as they activate against the stimulus itself and promote the activation of other cells, particularly macrophages via Th1 cytokines. During an immune response, professional antigen-presenting cells endocytose foreign material, which undergoes processing, then travel from the infection site to the lymph nodes. Once at the lymph nodes, the APC begin to present antigen peptides that are bound to class II MHC, allowing CD4 plus T cells that express the specific TCRs against the peptide/MHC complex to activate. When a Th cell encounters and recognizes the antigen on an APC, the TCR, CD3 complex binds strongly to the peptide MHC complex present on the surface of professional APCs. CD4, a CO receptor of the TCR complex, also binds to a different section of the MHC molecule. These interactions bring these proteins closer together, allowing the intracellular kinases present on the TCR, CD3 and CD4 proteins to activate each other via phosphorylation. With the assistance of a phosphatase present on the intracellular section of CD4-5, these molecules activate major Th cell intracellular pathways. These active pathways are known as signal 1 of T cell activation, as it is the first and primary pro-activation signal in a Th cell. Upon subsequent encounters with a given antigen, memory T cells are reactivated using the same TCR pathways. The binding of the antigen MHC to the TCR complex and CD4 may also help the APC and the Th cell adhere during Th cell activation, but the integrin protein LFA1 on the T cell and ICAM on the APC are the primary molecules of adhesion in this cell interaction. It is unknown what role the relatively bulky extracellular region of CD45 plays during cell interactions, but CD45 has various isoforms that change in size depending on the Th cell's activation and maturation status. For example, CD45 shortens in length following Th activation, but whether this change in length influences activation is unknown. It has been proposed that the larger CD45 may decrease the accessibility of the T cell receptor for the antigen MHC molecule, thereby necessitating an increase in the affinity of the T cell for activation. Once the activation has occurred, however, CD45 shortens, allowing easier interactions and activation as an effector T helper cell. Having received the first TCR/CD3 signal, the naive T cell must activate a second independent biochemical pathway, known as signal 2. This verification step is a protective measure to ensure that a T cell is responding to a foreign antigen. If this second signal is not present during initial antigen exposure, the T cell presumes that it is autoreactive. This results in the cell becoming anergic. Anergic cells will not respond to any antigen in the future, even if both signals are present later on. These cells are generally believed to circulate throughout the body with no value until they undergo apoptosis. The second signal involves an interaction between CD28 on the CD4+ T cell and the proteins CD80 or CD86 on the professional APCs. Both CD80 and CD86 activate the CD28 receptor. These proteins are also known as CO-stimulatory molecules. Although the verification stage is necessary for the activation of naive helper T cells, the importance of this stage is best demonstrated during the similar activation mechanism of CD8+ cytotoxic T cells. As naive CD8+ T cells have no true bias towards foreign sources, these T cells must rely on the activation of CD28 for confirmation that they recognize a foreign antigen. CD28 plays an important role in decreasing the risk of T cell autoimmunity against host antigens. Recognition. Once the naive T cell has both pathways activated, the biochemical changes induced by signal 1 are altered, allowing the cell to activate instead of anergize. The second signal is then obsolete. Only the first signal is necessary for future activation. This is also true for memory T cells, which is one example of learned immunity. Faster responses occur upon reinfection because memory T cells have already undergone confirmation and can produce effector cells much sooner. Once the two-signal activation is complete, the T helper cell then allows itself to proliferate. It achieves this by releasing a potent T cell growth factor called interleukin-2, which acts upon itself in an autocrine fashion. Activated T cells also produce the alpha subunit of the IL-2 receptor, enabling a fully functional receptor that can bind with IL-2, which in turn activates the T cell's proliferation pathways. The autocrine or paracrine secretion of IL-2 can bind to that same Th cell or neighboring Ths via the IL-2R, thus driving proliferation and clonal expansion. The Th cells receiving both signals of activation and proliferation will then become Th0 cells that secrete IL-2, IL-4, and interferon gamma. The Th0 cells will then differentiate into Th1 or Th2 cells depending on cytokine environment. IFN gamma drives Th1 cell production while IL-10 and IL-4 inhibit Th1 cell production. Conversely, IL-4 drives Th2 cell production and IFN gamma inhibits Th2 cells. It should be noted that these cytokines are pleiotropic and carry out many other functions of the immune response. In 1991, 3 groups reported discovering CD154, which is the molecular basis of T-cell helper function. Seth Laterman at Columbia University generated an murine monoclonal antibody, 5C8, that inhibited contact-dependent T-cell helper function in human cells, which characterized the 32-keta surface protein transiently expressed on CD4 plus T cells. Richard Armitage at Immunex cloned a cDNA encoding CD154 by screening an expression library with CD40Ig. Randolph Noel at Dartmouth Medical School generated an antibody that bound a 39-keta protein on murine T cells and inhibited helper function. Helper T cells are capable of influencing a variety of immune cells, and the T cell response generated can be essential for a successful outcome from infection. In order to be effective, helper T cells must determine which cytokines will allow the immune system to be most useful or beneficial for the host. Understanding exactly how helper T cells respond to immune challenges is currently of major interest in immunology, because such knowledge may be very useful in the treatment of disease and in increasing the effectiveness of vaccination. Proliferating helper T cells that develop into effector T cells differentiate into 2 major subtypes of cells known as Th1 and Th2 cells. Th1 helper cells are the host immunity effectors against intracellular bacteria and protozoa. They are triggered by IL-12 and their effector cytokines are IFN-gamma and IL-2. The main effector cells of Th1 immunity are macrophages as well as CD8 T cells, IgG B cells, and IFN-gamma CD4 T cells. The key Th1 transcription factors are STAT4 and Tbet. IFN-gamma secreted by CD4 T cells can activate macrophages to phagocytose and digest intracellular bacteria and protozoa. In addition, IFN-gamma can activate INOS to produce NOx free radicals to directly kill intracellular bacteria and protozoa. Th1 overactivation against autoantigens will cause type 4 delayed type hypersensitivity. Tuberculin reaction or type 1 diabetes belong to this category of autoimmunity. Verification. Proliferation. Th2 helper cells are the host immunity effectors against extracellular parasites including helminths. They are triggered by IL-4 and IL-2, and their effector cytokines are IL-4, IL-5, IL-9, IL-10, IL-13, and IL-25. The main effector cells are eosinophils, basophils, and mast cells as well as B cells, and IL-4/IL-5 CD4 T cells. The key Th2 transcription factors are STAT6 and GATA3. IL-4 is the positive feedback cytokine for Th2 cells differentiation. Besides, IL-4 stimulates B cells to produce IgE antibodies, which in turn stimulate mast cells to release histamine, serotonin, and leukotriene E to cause bronchoconstriction, intestinal peristalsis, gastric fluid acidification to expel helminths. IL-5 from CD4 T cells will activate eosinophils to attack helminths. IL-10 suppresses Th1 cells differentiation and function of dendritic cells. Th2 overactivation against autoantigen will cause TYPE1 IgE-mediated allergy and hypersensitivity. Allergic rhinitis, atopic dermatitis, and asthma belong to this category of autoimmunity. In addition to expressing different cytokines, Th2 cells also differ from Th1 cells in their cell surface glycans, which makes them less susceptible to some inducers of cell death. Effector function. Determination of the effector T cell response. Th1/Th2 model. Th17 helper cells. Th-alpha-beta helper cells. While we know about the types of cytokine patterns helper T cells tend to produce, we understand less about how the patterns themselves are decided. Various evidence suggests that the type of APC presenting the antigen to the T cell has a major influence on its profile. Other evidence suggests that the concentration of antigen presented to the T cell during primary activation influences its choice. The presence of some cytokines will also influence the response that will eventually be generated, but our understanding is nowhere near complete. Th17 helper cells are a subset of T helper cells developmentally distinct from Th1 and Th2 lineages producing interleukin-17. Th-alpha-beta helper cells provide the host immunity against viruses. Their differentiation is triggered by IFN-alpha/beta or IL-10. Their key effector cytokine is IL-10. Their main effector cells are NK cells as well as CD8 T cells, IgG B cells, and IL-10 CD4 T cells. The key Th-alpha-beta transcription factors are STAT1 and STAT3 as well as IRFs. IL-10 from CD4 T cells activate NK cells ADCC to apoptose virus-infected cells and to induce host as well as viral DNA fragmentation. IFN-alpha/beta can suppress transcription to avoid virus replication and transmission. Overactivation of Th-alpha-beta against autoantigen will cause type 2 antibody-dependent cytotoxic hypersensitivity. Myasthenia gravis or Graves' disease belong to this category. Limitations to the Th1/Th2 model. The interactions between cytokines from the Th1/Th2 model can be more complicated in some animals. For example, the Th2 cytokine IL-10 inhibits cytokine production of both Th subsets in humans. Human IL-10 suppresses the proliferation and cytokine production of all T cells and the activity of macrophages, but continues to stimulate plasma cells, ensuring that antibody production still occurs. As such, IL-10 is not believed to truly promote the Th2 response in humans, but acts to prevent overstimulation of helper T cells while still maximizing the production of antibodies. There are also other types of T cells that can influence the expression and activation of helper T cells, such as natural regulatory T cells, along with less common cytokine profiles such as the Th3 subset of helper T cells. Terms such as regulatory and suppression have become ambiguous after the discovery that helper CD4+ T cells are also capable of regulating their own responses outside of dedicated regulatory T cells. One major difference between regulatory T cells and effector T cells is that regulatory T cells typically serve to modulate and deactivate the immune response, while effector T cell groups usually begin with immune-promoting cytokines and then switch to inhibitory cytokines later in their life cycle. The latter is a feature of Th3 cells, which transform into a regulatory subset after its initial activation and cytokine production. Both regulatory T cells and Th3 cells produce the cytokine transforming growth factor beta and IL-10. Both cytokines are inhibitory to helper T cells. TGF-beta suppresses the activity of most of the immune system. There is evidence to suggest that TGF-beta may not suppress activated Th2 cells as effectively as it might suppress naive cells, but it is not typically considered a Th2 cytokine. The characterization of another novel T helper subtype, T helper 17 cells, has cast further doubt on the basic Th1/Th2 model. These IL-17 producing cells were initially described as a pathogenic population implicated in autoimmunity but are now thought to have their own distinct effector and regulatory functions. Of note, recent evidence suggests that functional plasticity is an intrinsic capacity of T helper cells. Indeed, a study in mice demonstrated that Th17 cells transform into Th1 cells in vivo. A subsequent study furthermore showed that extensive T helper cell plasticity is also prominent in man. Many of the cytokines in this article are also expressed by other immune cells, and it is becoming clear that while the original Th1/Th2 model is enlightening and gives insight into the functions of helper T cells, it is far too simple to define its entire role or actions. Some immunologists question the model completely, as some in vivo studies suggest that individual helper T cells usually do not match the specific cytokine profiles of the Th model, and many cells express cytokines from both profiles. That said, the Th model has still played an important part in developing our understanding of the roles and behavior of helper T cells and the cytokines they produce during an immune response. Recent scientific studies by Stockinger et al. Revealed that another T helper subset may exist. Th9 cells are claimed to be an IL-9 producing T cell subset focused on defending helminth infections. Memory T cell. Historically, memory T cells were thought to belong to either the effector or central memory subtypes, each with their own distinguishing set of cell surface markers. Central memory T cells reside in the lymph nodes while effector memory T cells lack the CC chemokine receptor type 7 and L-selectin receptors, which prevents them from trafficking to the lymph nodes. Additional populations of memory T cells are now known to exist. These include tissue-resident memory T cells and virtual memory T cells. The single unifying theme for all memory T cell subtypes is that they are long-lived and can quickly expand to large numbers of effector T cells upon encountering their cognate antigen. By this mechanism they provide the immune system with memory against previously encountered pathogens. Role in disease. Considering the diverse and important role helper T cells play in the immune system, it is not surprising that these cells often influence the immune response against disease. They also appear to make occasional mistakes, or generate responses that would be politely considered non-beneficial. In the worst-case scenario, the helper T cell response could lead to a disaster and the fatality of the host. Fortunately, this is a very rare occurrence. The immune system must achieve a balance of sensitivity in order to respond to foreign antigens without responding to the antigens of the host itself. When the immune system responds to very low levels of antigen that it usually shouldn't respond to, a hypersensitivity response occurs. Hypersensitivity is believed to be the cause of allergy and some autoimmune disease. Anti-tumor immunity. Hypersensitivity. HIV infection. Hypersensitivity reactions can be divided into 4 types. Other cellular hypersensitivities include cytotoxic T cell-mediated autoimmune disease, and a similar phenomenon, transplant rejection. Helper T cells are required to fuel the development of these diseases. In order to create sufficient autoreactive killer T cells, interleukin-2 must be produced, and this is supplied by CD4 plus T cells. CD4 plus T cells can also stimulate cells such as natural killer cells and macrophages via cytokines such as interferon gamma, encouraging these cytotoxic cells to kill host cells in certain circumstances. The mechanism that killer T cells use during autoimmunity is almost identical to their response against viruses, and some viruses have been accused of causing autoimmune diseases such as type 1 diabetes mellitus. Cellular autoimmune disease occurs because the host antigen recognition systems fail, and the immune system believes, by mistake, that a host antigen is foreign. As a result, the CD8 plus T cells treat the host cell presenting that antigen as infected, and go on to destroy all host cells that express that antigen. Some of this section is a simplification. Many autoimmune diseases are more complex. A well-known example is rheumatoid arthritis, where both antibodies and immune cells are known to play a role in the pathology. Generally, the immunology of most autoimmune diseases is not well understood. Perhaps the best example of the importance of CD4 plus T cells is demonstrated with human immunodeficiency virus infection. HIV mainly targets lymphoid CD4 plus T cells, but can infect other cells that express CD4 such as macrophages and dendritic cells. It has been proposed that during the non-symptomatic phase of HIV infection, the virus has a relatively low affinity towards T cells, resulting in a slow kill rate of CD4 plus T cells by the immune system. This is initially compensated for via the production of new helper T cells from the thymus. Once the virus becomes lymphotropic, however, it begins to infect CD4 plus T cells far more efficiently, and the immune system is overwhelmed. Of note, recent studies suggest that only 5% of the lymphoid-derived CD4 T cells targeted by HIV are permissive and become productively infected with the virus. More than 95% of the CD4 T cells that die are resting and are unable to support productive infection. These cells undergo abortive infection with HIV. Cell death is triggered when the host cell detects HIV foreign DNA intermediates and initiates a suicidal death pathway in an attempt to protect the host, leading to caspase-1 activation in inflammasomes, thus causing pyrotosis. At this point, chronic inflammation ensues, and functional CD4 plus T cell levels begin to decrease, eventually to a point where the CD4 plus T cell population is too small to recognize the full range of antigens that could potentially be detected. The depletion of CD4 T cells and the development of chronic inflammation are signature processes in HIV pathogenesis that propel progression to acquired immune deficiency syndrome. CD4 T cell depleted to the cell count of less than 200 cells/μl in blood during AIDS allows various pathogens to escape T cell recognition, thus allowing opportunistic infections that would normally elicit a helper T cell response to bypass the immune system. While these complete bypass situations only occur when the helper T cell response is absolutely necessary for infection clearance, most infections increase in severity and/or duration because the immune system's helper T cells provide less efficient immune response. Two components of the immune system are particularly affected in AIDS, due to its CD4 plus T cell dependency. If the patient does not respond to HIV treatment, they will succumb usually to either cancers or infections. The immune system finally reaches a point where it is no longer coordinated or stimulated enough to deal with the disease. Inhibition of CD4 T-cell expansion during HIV infection may occur due to microbial translocation in an IL-10-dependent way. Triggering PD-1 expressed on activated monocytes by its ligand PDL1, induces IL-10 production which inhibits CD4 T-cell function.